Good afternoon, everybody, and welcome to Journal Club. I'm in London today. I can hear Big Ben chiming, so that means it's 3 o'clock, and it's time to go. So today, Dr. Phil Norville is going to be leading us off to talk about environmental sampling of surfaces in a paediatric BMT setting. Phil, would you mind just advancing the slide? Great, thanks, Phil. So we talked about the aims of our journal club a little bit last time. What we really want to try and do is prompt a little bit of discussion to get people thinking. So that involves you. I hope you've had a chance to take a look at the paper, and we're hopeful that we'll get lots of questions in the Q&A. If I can just direct you towards the Q&A for your questions, and the chat for more general comments. I'll try and keep an eye on both. And I suppose in an ideal world, Phil will have a bit of fun. That's the plan. So without further ado, Phil, I'm going to hand straight over to you to kick us off.
Thanks, John. Yeah, just to build on what John said there, we had the first journal club last week, and we were really enthused by just how well it went and how many people attended. But yeah, we really do need buy-in from the audience in terms of the questioning to make the discussion go a bit further. We've got 45 minutes, so I'm probably only going to talk for 10 to 12 minutes on the paper and my findings, and then I guess it's over to you to ask questions. So I think it's a really nice place to start with why I chose this article. I'm fascinated by the role of surfaces in the transmission of healthcare-associated infections. I think it's amazing how well some of these organisms can survive in the environment and just for how long. But I'm also really interested to see what that means for our patients. So if these organisms are there, how do they get past the patient? How long can they stay around? Do they survive environmental decontamination? All kinds of questions like that. I also chose this paper because I've really enjoyed reading research from the authors previously. And I recommend that you have a look at the group because they've been quite active in their publication record, and they've gone and answered quite a couple of interesting questions around the methodology for sampling the healthcare environment and how it changes depending on the approach that you take. So we know there's a lot of variability in the sample technique that we use, and I wondered whether or not this paper might consider that, and also look about whether or not the techniques that they use are reproducible, because we know that the approach that we take will differ with the results that we're given. And also, I think it's a really interesting paper because I'm really interested on whether or not, if we're going to look at the healthcare environment and look for the organisms that are there, whether or not we do that routinely. At the moment, I think that we are quite reactive in the sense that we do it when outbreaks are ongoing and we're trying to identify the source of a particular outbreak. So what about employing that routinely? And I wondered whether or not that might be discussed within this paper. And also, we know these organisms are there, but are the methods that we're using to keep the environment clean and safe and effectively decontaminated, are they effective? So I'm going to talk a little bit about the method that they use for environmental decontamination, but how can we actually assess whether or not the interventions that are taking place are effective in decontaminating the healthcare environment? So loads of reasons why I chose this one. So if we get into the study itself, it was done at Great Ormond Street Hospital for Children in London, and it was done in an area of the hospital where some of the sickest and most vulnerable patients were. So a bone marrow transplant unit and haematology oncology unit. So if you think about the patient population there, very vulnerable, very young, and lots of challenges about keeping them safe and protecting them from infections. And what they were doing in that study was they were doing weekly sampling of 22 surfaces in communal spaces. And I think that's the first key point to get across, really, is this sampling was taking place in communal spaces. So looking in things like kitchens and equipment that's within kitchens. My experience of paediatric settings is quite often the family spend a lot of time in those settings. So there are things that are in place to make it feel a little bit more like a home and not a clinical setting. So they were looking at equipment, things like toasters and fridges, which you'll commonly find in some of these units. And they were doing this over a weekly period of 9 weeks. And they were collecting the samples with pre-moistened rayon floc swabs, which is essentially the name and what the swabs look like. And I think that's the first thing which piqued my interest and started the wheels turning about the fact that the swabs were pre-moistened, because I know that if you pre-moisten swabs, it's going to actually improve the efficacy of what you pick up from the environment. So again, already starting to think about how you vary your approach to sampling the environment and what results that it might give you. And in terms of what they were looking at aerobic colony count, so detecting the number of colonies that were growing after these swabs were streaked onto agar. And they were doing that for every 100 centimeters squared. And then they went one further than aerobic colony counts, and they were actually looking to the individual species identification using MALDI-TOF. And for this, where they couldn't get an identification via MALDI-TOF, they were doing sequencing of something called the 16S ribosomal DNA. So I've taken some images from the paper because I think it just really helps build a bit of a picture of what was going on. And you can see here on the figure on the right-hand side, the different sites within the communal settings that they sampled. And I think really it's quite obvious, isn't it, that these communal settings are kind of housed in that central location within the department there. So you can see they're kind of really nicely focused around that kind of main nuclei of the two units. And I don't know if you can make it out, but you can actually see how there's a split and how the corridor connects the wards between Ward 1, between the bone marrow transplant unit and the haematology oncology unit as well. So you can see there's that hub, which you can imagine being there. There's obviously a hive of activity around that focal site within the ward. People using, computers to type in data, maybe getting ready for ward rounds, having conversations. So I think that's why the communal setting is really important, because there's probably a lot of activity, a lot of staff come into contact with that environment and asking the question, what does that mean for contamination or the representation of these organisms in the healthcare environment. And there was a lot of variability in the surfaces that they analysed, and this is one of the reasons I really enjoyed reading the paper, was that they're looking at things that are commonly touched by lots of different people. So I just picked out a couple here, but toasters, fridge handles and keyboards are just some examples. If you think about that equipment, some of it's probably touched by just clinical or nursing staff, some of it's probably touched by parents and visitors and patients, and some maybe by both. So it's also really interesting to see, does that have an impact depending on where the item is or what it is on the diversity of the different organisms that it's contaminated with. And I think this is one of the key things for me as well that I was really interested in, was that this sampling was actually taking place after or during an adenovirus outbreak. So they were already using enhanced environmental decontamination measures. I don't think they mentioned in the paper what routine decontamination is. I think it might have been sodium hypochlorite at a much lower concentration. But I think this is one of the key things, is they were already doing enhanced environmental decontamination with 5,200 parts per million chlorine in a pre-impregnated wipe. So they were already doing something different. And what does that mean for environmental decontamination? You would hope, if you're doing enhanced cleaning, that it's going to reduce the bioburden. And they displayed their results as geometric means. So these are the key findings. The chart on the right is a real nice one because it just shows the diversity of the different microorganisms that were recovered, some of which I had to go on Google because I'd never heard of them before. But they collected a lot of samples over a 9-week period, and keyboards were the highest contaminated, so the highest geometric mean of aerobic colony counts per 100 centimeters squared. And if we just have a look at what that might be represented by, the organism which came out as having the highest percentage representation was an organism called Rhizobacter radiobacter, which I must confess I'd never heard of before reading this paper. And why that's really interesting is that it's commonly found in soils. Opportunistic pathogen, so can cause disease in vulnerable people, like the patients within this healthcare setting. But again, how is that being introduced into the environment? Is it coming in on visitors? Is a member of staff colonized? I thought that was really interesting, as an organism that you only tend to see in the soil, in the environment, being picked up in this paediatric unit. So keyboards were the highest level of contamination, kitchen microwave handles the lowest. Again, then just starting to look at some of the diversity, more gram-positive organisms than gram-negatives. So for those of you learning a little bit more about microbiology, gram-positives are things like Staph aureus, whereas gram-negatives are things like E. coli and Pseudomonas aeruginosa. So slightly skewed towards the gram-positives. And this is where it starts to get really interesting. Look at the diversity of the different species that were detected in this paper, 57 different species. And again, if you look at the chart on the right-hand side, you can just see how some of them are distributed. So if you'd asked me to name the organisms that I would have expected them to pick up, I would have gone for the organisms that we commonly find on our skin. So things like Staph epidermidis, Staph aureus, maybe some E. coli, etc. And some of those organisms are in there. But what was really interesting to me was that there was a lot of organisms that I wouldn't have imagined to be contaminating the healthcare environment, or maybe not contaminating the healthcare environment in such big numbers. And you can tell my enthusiasm gathering now. The other interesting thing is that the contamination, or what they detected in the environment, was quite transient as well. So this organism that they saw in a really high abundance, Rhizobacterium radiobacter, they only predominantly identified that on one day of the sampling that they did. So I'm assuming that on the other days, they didn't detect it in as high numbers, which I think is really interesting to think about, how these populations of organisms might be transient, and that they might be there one day and then a couple of days later they're not. Is that because they're displaced? Is it because the intervention is actually working and getting rid of the contamination? So again, really interesting to think about that and the impact on patients and healthcare associated infections. And as you'd expect, sites associated with frequent contact of staph had the highest bacterial counts and the highest bacterial diversity, so the highest number of different species. And just finally, some other key findings. So keyboards accounted for 3 out of the 5 most diverse sample sites during the study duration. If you think about keyboards, they're probably used commonly by lots of different members of staff, so probably unsurprising. And the diversity of these keyboards in terms of the organisms that were there were also the highest. So 2 of the keyboards had the highest level of microbial diversity, 28 different species on one and 21 on another. And again, I think that's something really interesting to think about from an infection control perspective, is that we've got all these different organisms there, but what does that mean for our patients or for the transmission of these organisms? Does a high diversity mean that we're more likely to get an infection because we've got more chance of picking up lots of different bugs from the environment? Or actually, does a high diversity mean that the numbers of these microorganisms are quite low because they're all competing with each other for space and nutrients? And actually, the numbers there mean that they're not high enough to cause disease or infection. So really interesting. I struggled a little bit then with some of the research that they'd done, or the analysis about grouping, but they'd basically classified these organisms by their ecological niche and put them into different grouping categories. And grouping sites by perceived contamination risk or possible transmission route to patients were not that good predictors of actual contamination, whereas actually grouping sites by activity, sample type, or predominant contact group was a better predictor of contamination. I've only really scratched the surface in terms of the key findings there. There's a lot more that's done in the paper, and I encourage you to go and read it. But I think, the key highlights really are that there is a lot of diversity in this environment. Keyboards are the most commonly or the most highly contaminated, and there's lots of diversity within those pieces of equipment as well. In terms of limitations, and the authors do a really good job of talking about some of these, that there could be variation in the cleaning efficacy. So some of the equipment that is being sampled was the responsibility of housekeeping teams to be decontaminated, whereas some was that of the nursing staff. And I think that might be a really interesting comparison. Is the efficiency of environmental decontamination higher in one group or the other? And what's the difference? And what does that mean for environmental contamination? Well, I'd like to see them look at the presence of multi-drug resistant organisms, because these are, again, what we might consider as being the biggest risk to our patients, because if they're picking up organisms that have resistance to antibiotics, then when they get an infection, they're obviously trickier to treat. I guess this is a limitation of not just the paper here, but of any environmental sampling, is this challenge of trying to analyze the presence of dry surface biofilms within the environment. Lots of studies now showing that dry surface biofilm are very prevalent within the healthcare environment, and there are lots of challenges around how you can actually detect dry surface biofilm in the environment. A lot of the work that I've read says that you need to focus on really high magnification electron microscopy to be able to see the biofilm in situ. But we know actually from published research that dry surface biofilm means that these organisms can really persist in the environment for long periods of time. So it would have been really interesting to see if there was dry surface biofilm at play here, and whether or not that accounted for some of the diversity that was present. And obviously, this was just looking at bacteria, so there was no analysis of viral contamination, which again, whether or not that is necessary is a good question. Viral contamination, assessing that is a lot more difficult, it's a lot more costly, and it's probably likely to be a lot more transient as well, because viruses don't survive in the environment for as long. So in terms of what that means for IPC, before we go to Q&A, not unsurprising really that keyboards came out as being the most heavily contaminated. If you think about keyboards in your own healthcare organization, how frequently are they decontaminated? I very much doubt they're decontaminated between every user. And I also have a question about whether or not if they were decontaminated with a really strong concentration of bleach, how long they would last for. So do they play a role in transmission? You know, it's probably unsurprising that they were heavily contaminated and amongst the highest diversity. But, you know, how can we effectively decontaminate them? I've seen some really interesting novel innovations around using things like ultraviolet light. But, you know, are they being decontaminated effectively? And whose role is to decontaminate them? Is it clinical staff? I suspect so. But, you know, who has responsibility for that? The other really interesting thing, you know, is that if we are looking for these organisms in the environment, is that being aware that this contamination can be transient, i.e. it can be there one day and it cannot be the next. Probably unlikely if you've got that in an outbreak setting, because you've probably got a continual source of transmission. But again, you know, we might see something in the environment one day and it not be the next. I think viral contamination is an interesting one, but again, it's whether or not that is worthwhile looking into because of the expense and the difficulty that comes with it. And I kind of touched upon this point earlier, you know, what about diversity of contamination in terms of the species level? If we've got a keyboard that's contaminated with 50 different species of microorganism versus a keyboard contaminated with one, does one pose a bigger risk of transmission to patients than the other? It's a really good question, I think. And also, there's a bit of an ongoing debate as whether or not sampling of the healthcare environment could be used routinely, or should it just be used in outbreak settings where you're trying to really get down to the fact about where there is transmission coming from. Obviously, there's lots of factors to consider with that, about how much it costs, who does it, who's responsible for it, if they got the resource to do that within the organization, or should actually be done as a more targeted approach. So I think, you know, lots of connotations here for IPC. I think the key one for me is around, you know, keyboards and potentially shared equipment that people are coming into contact with frequently. Do they present a transmission risk? I'd love to see a study which went to do effective decontamination of keyboards in intensive care or paediatric units and see if that made an impact on transmission of infections, for example. So, you know, some really interesting questions, but a really nice piece of work, just trying to highlight the challenges there are when it comes to analysis of the healthcare environment and the organisms that are there. So I'm going to stop talking. I'll hand you back to Jon, and then hopefully we can have a bit of a discussion and get people's thoughts and questions about what we've just talked about. Over to you, Jon.
Okay, thanks, Phil. Really great précis of the paper and put the key points across really, really well. I too found the grouping by different use and different type kind of confusing to interpret. So it's certainly worth some discussion on that. One thing I picked out during your presentation that I hadn't noticed before is even though there was no distinction between the perceived contamination risk as judged by the users and the mean colony count, there was quite a difference in the diversity by those different perceived risks. And I noticed that the items considered low risk had a much higher proportion of environmental organisms and the medium and high have the non-environmental organisms linked to them, so presumably more from a human background. And that felt like it could be significant in terms of how we interpret that grouping.
Yeah, that's a really good point, isn't it? I think those dynamics are really interesting, aren't they? Trying to understand where the contamination is coming from, but ultimately like where the risk is. That's a really good point.
So where do you sit then, Phil, on the methods and the lack of sampling for antibiotic-resistant bacteria? How much of a limitation do you think that was in the meaning and the impact of these findings?
It's a good question. I think that it would have been nice to see, the impact or the presence of MDROs, because I think they are the things that we are most concerned about when it comes to patient safety and patient outcomes. I think I viewed this paper really as kind of a really interesting insight just to show what is going on in the healthcare environment and look at just diversity and look at grouping those different pieces of equipment in communal areas by what exactly they're contaminated with. So I think that the key for me really and the key findings was all around diversity and just showing just how I was really surprised by just how diverse the level of contamination was on certain pieces of those equipment. So I think it would have been a nice to have and do some analysis around MDROs. But I think if you take me as a microbiologist and a purist, it's just really interesting to see like the level of diversity out there and wondering maybe, whether or not the techniques that were used and the sampling method that was used, what that meant for the results. But I think given the research that's been done by this group previously, they were really well placed to choose the right technique, which was going to give them the most kind of reliable or reproducible results.
Yeah, and it kind of de-risks the project from an impact point of view to not only focus everything on antibiotic resistant bacteria, because I've been on quite a few environmental sampling fishing trips where you come back and you haven't caught anything because you haven't identified any of that one antibiotic resistant bacterium that you're looking for as maybe a point source in a CPE outbreak. You take tons of swabs, loads of time, loads of effort, loads of cost. And actually, because they're all sampled using highly selective methods, you don't get anything from it. So I quite like this idea of looking at aerobic colony count, but especially the diversity index from the samples. I've got a bit stuck at what it means and how we interpret it. I think that's the really interesting thing to come out of this paper. I think the next step is to think, well, what are the risks attached to high and low diversity from an environmental sample?
Yeah, it's a really good point.
It'd be quite nice to look at correlating antibiotic-resistant bacteria contamination alongside diversity. Maybe some parallel sampling might tell us something in future.
Yeah, definitely. Your point around looking for things and not finding them is really relevant, isn't it? And I think that, again, the way that I'm looking at this paper is that it's really interesting to look at the dynamics and the transient nature of some of the contamination that they detected. And I think it's just maybe starting to unlock some of the thinking in my mind about how we might use this routinely. And I think one of the reasons why papers like this are so interesting is because there's just so much complexity associated with sampling the environment in terms of the methodology, when, where, how, who pays for it, etc. So I think it's really nice just to look at the dynamics that are involved and think about what that means in terms of risk to the patients as part of that.
Yeah, okay, great. Let's go to the Q&A. Thanks to everyone who's put questions in. Please do fire away and get more into the Q&A box. A couple of people have mentioned hand hygiene, what the implications for hand hygiene these environmental sampling findings have.
Well, I think this is the key, isn't it? Is that we think about surface contamination and you might be, wow, 57 different species identified. But ultimately, the organisms have got to get from the surface to the patient, haven't they? So it might mean that there are failures in the way that we keep the environment safe. But ultimately, if we can wash hands and have a really good level of compliance for hand hygiene, then it can reduce the risk, can't it? So I think that it's really interesting to know that these organisms are there. It's fascinating to know the diversity that they're there. And There is obviously risk of then picking them up on your hands if you're coming into contact with it. But ultimately, good hand hygiene practice can eliminate some of that risk, can't it? So I think that if anything, this probably just highlights the importance of hand hygiene, because these organisms are there in the environment. We know that the hands of staff and patients are really good ways of transmitting these organisms from contaminated surfaces to themselves, and being really good vectors. So I think that hand hygiene really is crucial. And I think, just quickly, the other thing which I've seen a real kind of focus on with infection prevention the last maybe 18 months to 2 years is the role of patient hand hygiene. I think with patients who are spending a long period of time, bedbound and having difficulty in using sinks or having to use commodes for toileting and don't have the facility to wash their hands on a regular basis, I think trying to think about how we can incorporate hand hygiene for them via things like, obviously, the sustainability agenda around wipes and things like that. But I think we know that hand hygiene really is so good at reducing the risk of transmission. So we need to think about patient hand hygiene and providing or promoting it more. So promoting the patients who can wash their hands, doing it more effectively and more frequently. But also, what about the patients who can't, and giving them alternative methods?
I've certainly heard it said before, probably a few years ago now. I haven't heard it so much recently, that who cares about a contaminated environment? Doesn't matter what diversity, doesn't matter what AMR bacteria there. If we always washed our hands every time before patient contact, then it doesn't matter. It wouldn't be important in transmission. Is that something you recognise?
I think it certainly carries weight. But if you work in a healthcare environment, that isn't always possible. I think if you think about things like the 5 moments from WHO, that you could be washing your hands every minute potentially, and it's whether or not that's feasible. So it definitely carries weight, and I think that hand hygiene is probably the biggest intervention there is in reducing the risk of infections and transmission. But ultimately, we've got to be realistic, haven't we? And we know that people in healthcare environments, particularly in the UK, there's pressure around time. There's pressure around, ensuring that we're giving patients the care that they need, and there's pressure on the service all of the time. And we wash our hands when we can, but ultimately, there are things that will happen within the healthcare that we provide, which mean that we're not always doing it as frequently as we can, or there are barriers to us doing it. So, I definitely think that hand hygiene is really, really important and crucial. But, we know that hand hygiene alone isn't part of an effective IPC strategy. We need other interventions. And the way that I like to think of it is that it's a cumulative effect. So if we can reduce contamination in the environment, if we can isolate our infected or colonized patients, and we can wash our hands on a regular basis, I think we've got a much better chance of reducing infection, as opposed to just doing one of them on its own.
Yeah, I agree with you, Phil. I think I've certainly seen quite a lot of studies, and especially the DNA marker studies that present this picture of incredibly rapid and dynamic transfer of, in that case, a model organism, but probably microorganisms too, around the healthcare environment, bouncing around from surface to hand and hand to surface. It's really difficult to disentangle the relative importance of various transmission routes. But one thing we do know for sure is that the psychological trigger to wash your hands is much weaker after touching a surface than after touching a patient. We know that all these kinds of self-protection triggers are going on in our minds. So I've always thought that the contamination of surfaces is in some ways going to be relatively more important, because you're less likely to wash your hands after touching a surface than after a patient. Lots of love, by the way, for your mention of patient hand hygiene. Definitely a future topic for a journal club, I think, to look into a bit of more detail about how we can involve our patients in hand hygiene. Okay, let's go down a slightly microbial route. A couple of comments around sampling methods, and particularly dry surface biofilm. So you mentioned that this study didn't include any reference to or mention of dry surface biofilm, but how would a study do that? How could a study do that?
The ones that I've seen previously have looked at using electron microscopy. There's a really nice study done by the group at Cardiff in Jean-Yves Maillard's group at the School of Pharmacy, just essentially assessing a bit of an extension of what they've done here, looking at routine swabbing and seeing if they could detect the presence of dry surface biofilm or even microorganisms. And I think that they actually cultured nothing from all of the sample sites that they analysed, and this was done over 3 different geographically distinct hospitals, lots of different variations in the equipment that they sampled, and their swabs were coming back negative. The only way that they can actually go and detect dry surface biofilm was chopping up and using really high electron microscopy to visualize the biofilm in situ on these environments. So not something that just is sitting down the corridor from the IPC office, unfortunately, an electron microscopy suite, which costs a couple of millions of pounds. I've seen some really interesting, research and concepts about potentially using some kind of dye or reaction which will actually change color when it comes into contact with things like the exopolysaccharide that is in dry surface biofilm. So this is kind of like the dehydrated layer that protects the organisms from desiccation and disinfectants. So could we have something that potentially changes color when it reacts with components of that EPS? But at the moment, I think it's still early stages. I think we're still kind of uncovering the significance of dry surface biofilm and the role that it plays, whether or not we're going to have a methodology which is able to detect it in real time. The other study actually that I've heard of recently is using Raman spectroscopy to detect biofilm in situ, but I think that's really early stages. So I think it's a really exciting area of IPC that we need to pay more attention of. And for me, it's probably the reason why we see such diversity and why some of these organisms can survive for such long periods of time. My personal view is I think it's down to dry surface biofilm. So watch this space for more techniques around shining a light on dry surface biofilm.
Yeah, it's interesting. I was one of the 3 study sites for that Jean-Yves study. So I went around collecting up loads of keyboards and sent them off to the lab in Cardiff to be chopped up and put under the electron microscope. I mean, it's never going to be a high throughput, low cost method, definitely not. But the findings were pretty surprising. As you mentioned, we didn't culture anything really significant, but in the dry surface biofilm, there were viable, really significant organisms, quite a high proportion of MRSA, which we didn't see much of in the water at that time. So I think it's almost like the rings of a tree harboring older microbes, and a bit of an open question as to how much of a risk the bacteria buried deep in the biofilm are, but I think there's probably enough to suggest that they get disrupted and slough off, such that those organisms buried deep in the biofilm are at least potentially going to come to the fore and be involved in cross-transmission.
Yeah, definitely. And I think this is one of the reasons why the healthcare environment to me is so fascinating. If those organisms are there dormant and they're just sitting there in wait, what happens if we actually reactivate them, and what kind of things that we do in the environment do reactivate them? Does washing or decontaminating a surface with a neutral detergent mean that actually you remove that top layer and you actually get access to the biofilm, and then you kind of reseed the environment? So does dry surface biofilm act as this kind of reservoir? So I think it's a really interesting thing and a really interesting topic of research is, if these organisms are there and they're surviving, how do they kind of come back to life? What is it that happens that mean that they are able to go and cause an outbreak or reactivate to cause disease?
Okay, just before we move away from biofilms, what about our choice of cleaning disinfection in the context of a dry surface biofilm? How does it influence our choices around cleaning and disinfection methodologies?
How long's a piece of string? I think, again, it's a really interesting topic of research. And I've seen studies showing that you can use 20,000 parts per million chlorine. So a concentration that we're probably never ever going to routinely employ within healthcare. No, we're not OE because it's 20 times stronger than what we recommend in the UK. And you still get dry surface biofilm coming back. The big thing that I've seen with dry surface biofilm is that you can think that you hit it. So you get down to, okay, maybe not being able to culture it anymore. But as time goes on, the biofilm essentially comes back because you've always got that kind of residual reservoir. So I think we need to think a little bit differently about environmental decontamination when it comes to biofilm. I think we probably need to think maybe more about what's the impact of the actual mechanical removal. So if we're thinking about wiping a surface, do we need to think more about the components that make up that substrate? And do we need something which is maybe slightly more abrasive? Not that it damages the surface, but it actually maybe has some mechanical abrasive action in breaking that biofilm layer. And ideally, I would like to see that combined with the disinfectant. So if you can actually break down the structure, leave something that's left behind on the surface, which has disinfectant properties, ideally something a bit stronger. So something like a high powerful oxidizing agent, I think would be my kind of method of choice. I would be thinking about, if I was decontaminating dry surface biofilm, throwing a little bit more at it than routine environmental decontamination practice, because we know from the research that these organisms are quite hardy when they're in a dry surface biofilm state. But I think, this is why it's so interesting. Innovation in this space, thinking about novel methods, novel substrates for wipes and things like that, is a real way that we can kind of engineer new techniques to tackle these challenges.
Yeah, agree. And I did a bit of research years ago now looking at dry surface biofilms and disinfection. And the higher the reactivity of the disinfectant, the more chance there was to not only inactivate kind of the surface microbes of the biofilm, but begin to disrupt the biofilm matrix and then get to the lower portions of the biofilm. So things like hydrogen peroxide, peracetic acid, chlorine, those oxidizing chemicals seem to do a better job of getting deeper into the biofilm.
Mm-hmm.
Okay. What next? There was a great comment in the chat about our discussion about antibiotic resistant organisms. MDROs are like fancy cars in IPC that grab all the attention. But actually, the vast majority of healthcare-associated infections not caused by antibiotic-resistant organisms, caused by antibiotic-susceptible versions that are just as transmissible, and these infections are very often endogenous. So we need to focus as much on preventing endogenous infection as transmission of antibiotic-resistant bacteria. So good comment. Thanks for that.
I love that one.
Okay.
And I'd like to think maybe that these organisms are just adding on little upgrades as they go along through life. So you're putting a sports package on your VRE, which means you're resistant to colistin or something like that. I think that's a really nice analogy.
Yeah, we could definitely develop that one. Okay, so let's focus in on keyboards then, because that seemed to be the predominant theme coming out of this evaluation. I think you said keyboards accounted for 3 of the 5 most contaminated
Correct.
items. They also had really high diversity, which, as you said, is not a surprise given all the grubby mitts that are out there touching them day in, day out. What do we do about that practically? How can we improve keyboard hygiene? On the basis of these findings, do we need to improve keyboard hygiene?
It's a good question. I think, given the level of diversity that's there and that they are amongst the most highest contaminated equipment isn't surprising. I think, the real challenge is just how frequently these things are used, and that how environmental decontamination of them is going to fit into their routine use. If we're wiping a surface and then somebody's coming back to it and using it before that has dried, what impact does that have on the environmental decontamination procedure? I think where I would have loved this study to go next, potentially, would be to see, is there any transmission from these keyboards to patients, and is there actually, a route? You would probably suggest there potentially is, given that we're proving how highly contaminated and how low the level of diversity is on them, and the fact that they are probably being touched by clinical staff really frequently, and then they potentially go to a patient. I'm all ears really as to what we could do to decontaminate our keyboards more effectively. And again, I think you could do a whole new study to just look at how the population of these microorganisms on keyboards changes. I mean, I suspect that it changes quite frequently depending on the dynamics within the organisms that are there. Do organisms come in and replace ones that are there? What happens if you do wipe it and it becomes contaminated? How quickly does it become recontaminated? Do we put all the keyboards into a ultraviolet light cabinet at the end of the day, and reduce our risk that way? I mean, our healthcare organizations are 24/7, so the chances of doing that are really slim. So yeah, what do you think, John? Is there any kind of room for improvement about how we can effectively decontaminate kit like that?
I think there's definite risk. There's some compelling data that keyboards become contaminated with really significant organisms. There's some intervention studies coming through showing that if we do a better job of cleaning them and related equipment, we can reduce risk of transmission. I'm just trying to think if I've ever seen anybody in a clinical environment actually clean a keyboard. I have, but it was during the COVID years. I just don't think they're cleaned by anyone hardly any of the time. So I think there's massive room for improvement. Somebody in the chat suggested implementing some kind of ATP bioluminescence-based method to assess cleaning of the keyboards. I think that makes sense. We can think about fluorescent markers. Design opportunities as well. Some designs are definitely better than others. If you go and have a look at your standard QWERTY with all the gaps down it, it's going to be pretty much impossible to clean. There's certainly some keyboards out there with a better design, but I think more work to be done on getting the design optimized.
Yeah, and as well as the design, it's whether or not they can withstand what we're throwing at them, isn't it? I think I share what you just said about whether or not I've actually seen somebody clean a keyboard before. So if we are going to start cleaning them, can they withstand what we're throwing at them? What does that mean for their integrity? And there's quite a lot of work now being done about the importance of material compatibility, given how often we disinfect surfaces or we decontaminate them. And when we're throwing harsh chemicals at them, what does that mean for equipment? And what about some of this really expensive equipment that we decontaminate as well? So I think there's definitely a role for collaboration there as well between manufacturers of decontamination products and equipment and medical devices within healthcare.
Yeah, definitely opportunities for some of the things mentioned in the chat, the Q&A around flat keyboards and maybe antimicrobial surfaces as well. Somebody's mentioned mobile phones. I'm not sure we've got time or capacity to think about mobile phones, but it's similar, isn't it? Because they are absolutely ubiquitous now out there in the clinical environment. So one observation that I've had recently is we in IPC spend a lot of time auditing. We ask people to do a lot of auditing as well of hand hygiene, vascular lines, urinary catheters, but not really shared medical equipment. We have routine audits in place for the kind of hard surface cleaning, but I wonder whether there's a bit of a gap in our set of interventions around a specific audit aimed at these kind of in-betweeny pieces of mobile medical equipment that aren't the responsibility of the cleaning and disinfection people to disinfect, but tend to fall to the actual clinical users, and do a bit of observation data and just say, look, 10% of these were actually met cleaning opportunities, 90% were missed.
Yeah, definitely. And just off the back of that, I completely agree. There's work that Brett Mitchell has done, the CLEAN study, where they've looked at specifically targeting shared medical equipment. So actually providing extra resource to the decontamination of that equipment. And I believe that the data does show it actually crosses over to a risk in transmission of infections. So I think that the more evidence that we can build around the role of shared medical equipment in the transmission of infections, the more that we can get the key people within the healthcare organisation to take notice about the risk, and then linking that to patient outcomes and risk is the next step. But yeah, I definitely think there is something that we can be done to look at more around our compliance. And why not start with the highest risk? Why not start with shared medical equipment, which we think is going to be the biggest risk? Because obviously there's a lot of shared medical equipment within our healthcare organisations. If we need to start somewhere, why don't we start with the stuff which holds or possesses the biggest risk?
Okay, thanks, Phil. Look, I'm really sorry to say we're right in the middle of discussion, but we are out of time for today. We didn't even get a chance to talk about some of the really important themes arising from the work around cost effectiveness, routine screening, and laboratory methods. We'll have to save those for another time. So just to let you know, in 2 weeks' time, we've scheduled our next Journal Club on Wednesday, the 31st of July. We'll have a special guest to present, Dr. Judy Robinson. We'll be talking about real-world observations to prevent SSI, and Phil's going to be facilitating the Q&A that day. Just to note, there's a website link there, and it's in the chat with the schedule for future journal clubs, which we'll be updating as they go live. And you can subscribe now to a list so that you get an email every time there's a journal club planned. And just finally, thanks to IPC Partners for hosting today. And most of all, thank you to you for joining us and for all your great interactions. Great to see you. Thank you.